Hello students, today we will have lecture on antigen. So now we will see definition of antigen. So antigen is defined as any substance that satisfies two immunological properties, that is immunogenicity and antigenicity. So what is immunogenicity? It is ability of antigen to induce immune response in body. And what is antigenicity? It is ability of antigen to combine specifically with antibodies or T cell surface receptors. Then we will see epitope. So what is epitope? It is smallest unit of antigenicity. It is also called as antigenic determinant. And there are two types of antigenic determinants. One is sequential or it is linear and it is mostly seen in T cells. And second is conformational or non-sequential epitope. So it is mainly associated with the B cells. Types of antigen. So there are two types, complete antigen and incomplete antigen. So complete antigen, when it induces antibody formation, it is called as complete antigen, and incomplete antigen is also called as hapten. So it is incapable of induction of antibody formation. Now we will see incomplete antigen, that is hapten. So definition is they are low molecular weight molecules that lack immunogenicity but retain antigenicity. So there are two types again. One is simple hapten and second is complex hapten. So simple hapten is univalent, contain only one epitope, that is antigenic determinant, and complex hapten, it contains two or more epitopes, and haptens can become immunogenic when combined with larger protein molecule called carrier. Means they can produce antibody when they combine with the other protein molecules. Now we will see antigen-host relationship. So it is self or autoantigen, so belong to host itself. So that is that antigen is present inside the host body. Then non-self, or it is also called as foreign antigen. That means it is present outside the host body. Then they are again of another different types. So first is alloantigen. So it is species specific antigens are called as alloantigen. This is when the antigens present in a species. Then isoantigen, present only in subset of species. Example, blood group antigen, histocompatibility antigen. So they are called as isoantigen. Then heteroantigen. So antigens belong to two different species. Example, heterophile antigen. Now we will see in detail about heterophile antigen. So what is definition? It is type of heteroantigen that exists in unrelated species, that is different species. Antibody produced against antigen of one species can react with other. So the mechanism is used as a diagnostic application in few tests. So example of that is first is Will-Felix reaction. So it is used for the diagnosis of typhus fever. So in that, antibodies against rickettsial antigen are detected by using cross-reacting protease antigens. Then second test is Paul-Banel test. This is used for the diagnosis of infectious mononucleosis. So in these, antibodies against Epstein-Barr virus, which is causing infectious mononucleosis disease. So these Epstein-Barr virus antigen are detected by using cross-reacting sheep RBC antigens. So these are two examples of heterophile antigen. Now we will see determinants of antigenicity. So what is antigenicity? We already saw it is property that makes substance antigenic. That means it combines with specifically with the antibodies. So there are factors depending on that the substance can become antigenic. So first is size. So large molecular weight, they are highly antigenic, and low molecular weight substances, they are less antigenic. Then second is a chemical nature. So proteins, polysaccharides, they are highly antigenic, and lipids, nucleic acid, they are less antigenic. Then third factor is a foreignness to host. So it is non-self. So they are immunogenic. Self, mostly they are not immunogenic. Then genetic factors. Then next is the optimal dose of antigen. Then next is the route of administration of antigen. So orally if you give antigen, then there is production of IgA antibody. In inhalation usually there is production of IgE antibody, and parenteral, that is intramuscular or IV, it is production of the IgG antibody. Then site of injection. So hepatitis B injection should be given in deltoid region because gluteal fat is there, and in that absence of APC means antigen processing cells are absent in that. So hepatitis B injection should be given in deltoid region. So site of injection is important factor. Then repeated dose of antigen. So they increase immune response. Then multiple antigens. So immune response is increased. In that adjuvant like action is there. Example of that is DPT vaccination. So in that pertussis act as an adjuvant. Then effect of prior administration of antibodies. So immune response is decreased. So example of that is RH immunization. So in that, if the Rh antibodies, the Rh immunoglobulin is given, then Rh antibodies are not produced, and then that is used in the treatment of the or prevention of the hemolytic disease of newborn. Now we'll see biological classes of antigens. So there are two types mainly depending mechanism of inducing antibody formation. So first is T cell dependent antigen. It is also called as TD. An example, all normal antigens are TD or T cell dependent antigens. Then second is T cell independent antigen. So it is also called as TI. So example, bacterial capsule, flagella or LPS, that is lipopolysaccharide. They are T cell independent antigens. And third type is super antigen. Now we'll see super antigen in detail. So definition, they are protein molecules that activate very large number of T cells irrespective of their antigenic specificity, and then they can cause disease. So we'll see examples of the super antigen and disease. So first is Staphylococcus aureus. So it has that enterotoxin which acts as a super antigen, and then it gives rise to disease like food poisoning, toxic shock syndrome. Then second example is group A Streptococcus. In that pyrogenic exotoxin is a super antigen, and then it causes disease like rheumatic heart disease. Then third is Epstein-Barr virus. In that Epstein-Barr virus antigen acts as a super antigen, and then it causes disease like B-cell lymphoma. Now we'll see in short about sequestrated antigen. So what is the definition? So certain self antigens are present in closed system and are not accessible to immune apparatus and are called sequestrated antigens or hidden antigens. So that means they are present in the body but they are in hidden form. So when they get exposed, then they cause reaction and then disease. So they are called as sequestrated antigen. So examples of few sequestrated antigen is first is lens antigen of eye. So it is enclosed in a capsule. So but while doing cataract surgery, if antigen leaks and then it causes immune response and then damage the lens of other eye. Then second is a sperms. So they are in a hidden form. But if mumps infection is there and then in that the leak of the sperms is there, then that causes immune response and then that causes orchitis, that is inflammation of the testes. Then third example is thyroglobulin. So it is present in hidden form, but antibody to thyroglobulin is formed and then that can cause disease like thyrotoxicosis or Graves' disease.
These are my references. I think you can study from these references and I hope you find this session useful. Thank you.